Welcome to Simply Micromed, bacterial cell or structure of bacteria. In this session, we will discuss about the structure and function and importance of bacterial cell or structure in detail. Bacterial cell, or we can also call it as structure of bacteria. And this is the picture of bacterial cell or structure of bacteria. And here you can see this is the outermost layer of bacteria, that is capsule. And all the organism will not have this capsule. Only the capsulated organism, we can see this capsule. And just inner to the capsule is the cell wall. And cell wall will be seen in all organism, and depending upon the organism, mainly gram positive and gram negative organism will have different type of cell wall. And just inner to the cell wall, we can see this is the cell membrane or cytoplasmic membrane. And inner to the cytoplasmic membrane, we will have this cytoplasm. And this cytoplasm consists of granules, nuclear material, that is double-stranded, linear or circular DNA can be seen, mesosome and ribosome and other organelles or plasmids, and everything can be seen in the cytoplasm of this bacteria. And also here you can see this is the hair-like structure. We can see the outermost layer, we can see the hair-like structure, short hair-like structure, that is called pili. This pili is for adhesion of bacteria to the host cell membrane. It will help in the virulence factor of the bacteria. And also here you can see this is the flagellum. Flagellum is a locomotory organ for the bacteria. This will help for the movement of bacteria or motility of bacteria. With the help of this flagellum, bacteria can move from one place to another place. And this is the anatomy of bacterial cell. Here you can see this is the outermost polysaccharide capsule. Just inner, you can see this is the cell wall. And just inner to the cell wall, we have cell membrane or cytoplasmic membrane. And in the cell membrane, you have the cytoplasm, and this cytoplasm consists of ribosome, bacterial chromosome, mesosome, plasmid, and other organelles. So cell wall. Cell wall, we already told that cell wall is the outermost layer for bacteria, not the outermost layer. And capsulated organism will have the capsule is the outermost layer. In cell wall, generally we will call it as the cell wall is the outermost layer or the tough and rough structure that will cover the bacteria. And the cell wall will give the rigidity and shape, so that means the cell wall will give the strength and shape of the bacteria. And also it will have a role in the virulence factor and immunity. Virulence factor will help in the pathogenesis, or it will also help in the infection causing. And it will maintain the osmotic pressure. And due to the maintaining the osmotic pressure, the bacteria will get the nutrients from outside the cells. And this is the site for phage absorption. And also it will take part in the cell division. The cell wall will be take part in the cell division. And this cell wall can be demonstrated by plasmolysis. Plasmolysis is the process by which we can detach the cell wall from the bacteria. Bacterial cell wall. This bacterial cell wall is made up of mucopeptide or peptidoglycan layer, and this is the structure of peptidoglycan layer. Here you can see this peptidoglycan layer is composed of alternate unit of N-acetyl acid and N-acetyl glycosamine. And here in this first is this N-acetyl glucosaminic acid. Next N-acetyl glucosamine. Again you can see this is N-acetyl acid. That is the alternative unit of N-acetyl acid and N-acetyl glycosamine. And this N-acetyl muramic acid is connected by a peptide bridge. Here you can see here, everywhere you can see this peptide bridge. And these two peptide bridge are connected by this pentapeptide bridge. So this is the structure of mucopeptide or peptidoglycan layer of bacterial cell. That is, it is connected. It is made up of alternative unit of N-acetyl acid and N-acetyl glycosamine, and this N-acetyl acid is having a peptide bridge, and these peptide bridge are connected by a pentapeptide bridge. Difference between gram-positive and gram-negative bacterial cell wall. And this is the picture of gram-positive bacterial cell wall and gram-negative bacterial cell wall. Here you can see this is the peptidoglycan layer of gram-positive bacteria, and this peptidoglycan layer consists of teichoic acid and lipoteichoic acid. And in gram-positive bacteria, you can see the thick peptidoglycan layer when comparing to the gram-negative organism. Here, this is the peptidoglycan layer of the gram-negative organism, and this one is the peptidoglycan layer is the of the gram-positive organism. Here you can see the thick peptidoglycan layer with teichoic acid and lipoteichoic acid, and this is the thin layer of peptidoglycan layer, and also this teichoic acid and lipoteichoic acid is not seen in this gram-negative organisms. And this is the cell membrane. And here you can see this is the cell membrane of the gram-negative bacterium. And in gram-negative bacterium, we will have the outer membrane layer. This is the outer membrane layer. In the case of gram-positive organism, doesn't have this outer membrane layer. And in gram-negative organism, we have outer membrane layer, and this outer membrane layer will consist of uh, contain phospholipid and lipopolysaccharides. This is lipid-rich lipopolysaccharides can be seen in outer membrane, and also this outer membrane will have small hole-like structure called porins. These porins can be seen in the outer membrane layer of gram-negative organism. And also you can see this is the periplasmic layer. This periplasmic layer can be seen in gram-negative bacterium, and mainly this periplasmic space will be generally not seen in gram-positive organism, but in some organism we can see in generally the periplasmic layer will be absent in gram-positive organism and comparing to this gram-negative organism. And this is the electron microscopic picture of gram-positive and gram-negative bacteria. Here you can see this is the peptidoglycan layer. This is the thick peptidoglycan layer can be seen in gram-positive bacteria. Here you can say this is thin peptidoglycan layer, and the outer membrane can be seen in gram-negative bacteria, but not in gram-positive bacteria. So periplasmic space, this is the periplasmic space, and this periplasmic space can be seen only in this gram-negative bacteria, not in this gram-positive bacteria. Cytoplasmic membrane. The cytoplasmic membrane, we already told that the outermost layer we have capsule, only the capsulated organism we can see this capsule. Just inner to this capsule we have bacterial cell wall, and inner to this cell wall we have this cell membrane or this cytoplasmic membrane. And the cytoplasmic membrane is a thin, elastic, semi-permeable layer. It is very thin. It is measured in 5 to 10 nanometer in size, and this elastic semi-permeable layer. The semi-permeable layer will help in the uh, control of inflow and outflow of metabolites, mainly by osmotic barrier. And it will maintain the osmotic barrier, and it will control the inflow and outflow of metabolics. That means bacteria doesn't have the mouth like us. So the nutrients will be entering to the bacteria due to this change in the osmotic pressure, and also the waste products of the bacteria will be go out through this cytoplasmic membrane, and this is controlled by this cytoplasmic membrane, and it is composed or made up of phospholipid and protein. Next, about the mesosome. Mesosome is a complex infolding of bacterial cell membrane, which is seen in bacterial cell membrane, and this is the center of respiratory activity, and also it is take part in the cell division. You can see this based upon the location. This mesosome will be divided into central mesosome and peripheral mesosome, and the central mesosome will help in the DNA replication and also the transfer of DNA. And this is the peripheral mesosome. It is also called the lateral mesosome. This seen in the lateral or peripheral side of the bacterial cell or the cytoplasmic membrane, and it is functionally similar to the mitochondria site of respiratory. That means it will help in the respiratory process of this bacteria. Cytoplasm. Cytoplasm is a colloidal system containing organic and inorganic solution. It contains ribosome, mesosome, inclusion granule, and vacuole. And this cytoplasm is seen inside the cytoplasmic membrane. So it's a colloidal system, contain organic and inorganic solution, and all the organelles of bacteria can be seen in the cytoplasm. Ribosome. Ribosome is a tiny granules present in the cytoplasm. They are globular structure composed of RNA and protein. They are made up of two subunits. For the larger one, subunit is 15S, and the smaller subunit is 13S. It measures 10 to 15 nanometer in size with sedimentation coefficient of 17S. The function of this ribosome is the function site of protein synthesis. The protein synthesis are take place inside this ribosome. Inclusion granules. These are non-living bodies deposited or seen in the cytoplasm. Inclusion granules will also, we will call it as inclusion bodies. And these are not permanent or essential structure. That means, and this is not always seen in all type of bacterial cell, only when they see the large amount of nutrients are present and disappear under the condition of starvation. That means when there is a large amount of nutrients are present in the bacteria, so this inclusion products can be seen, and when there is a depletion of the nutrition, this inclusion granules or body will be disappear from the bacterial cell. The example for this inclusion granule are lipid granule, volatile granule, and sulfur granules. And these inclusion granules are generally we can call it as a source of stored energy and that are present in some species of bacteria and that are also not always seen in bacterial cell wall or bacterial cytoplasm, not cell wall, bacterial cytoplasm. Nucleus. Nucleus in the bacteria is not well developed because the bacteria is a prokaryotic organism, not like eukaryotics. That means it doesn't have true nuclei. And this nucleus of the bacteria is a single or circular double-stranded DNA which is seen inside the cytoplasm of the bacteria. And this is controlled, the nucleus or the double-stranded DNA will control the growth and metabolism and multiplication of bacteria, and also it will hereditary transmission of character. That means the DNA it will carry the hereditary transmission of the character is by this double-stranded DNA seen in this nucleus. And this nucleus can be called as genophore, nuclear body, nucleoid, and chromosome. The plasmid are extracellular genetic material. It is also called as episome. This plasmid is also seen in the cytoplasm of the bacteria. It is extracellular genetic material. It is also a DNA, you can say the DNA, and it is responsible for drug resistance and toxigenicity of the bacteria. That means this plasmid doesn't have any relation or it will not control the growth and metabolism multiplication of bacteria and it is only responsible for drug resistance and toxigenicity seen in the bacteria. Capsule and slim layer. Capsule or slim layer is a gelatinous amorphous viscid layer seen in this outermost layer of capsulated organism. That means the slim layer and capsule is only seen in capsulated organism, not in all the organs. When it loose and irregularly arranged, we will call it as slim layer. When it is organized and sharply defined structure, we will call it as capsule. And the difference between slim layer and capsule is when it is loosely arranged or irregularly arranged, we will call it as slim layer. And it is sharply and it have a well-defined structure, then we will call it as a capsule. And this capsule or the slim layer will have this antigenic properties. That means it can stimulate the antibodies, and this capsule will protect the bacteria from phagocytosis. Phagocytosis means engulfing of the bacteria by the macrophages. It's called as phagocytosis. Due to this phagocytosis, the bacteria can be lysis or killed by these macrophages. And these capsulated organisms can be seen by negative staining and Quellen reaction. This is the negative staining picture of capsulated organism, and this is the unstained area. You can see this is the capsule, and just inner to this capsule we can see the bacterial cell. It is about the capsule and slim layer.
Flagella. Flagella is a thread-like structure from cytoplasm. It is organ for locomotion, composed of flagellum and also have an antigenic property. The flagella means all the bacteria doesn't have this flagella. Only the motile organism we can see the presence of this flagella, and this is an organ for locomotion. That means the bacteria will move from one place to the another cell or another place by the help of this flagella. So it will have importance in the virulence factors of the bacteria, and also it is have antigenic property, and it is composed of flagellin. And now you already told that all the organism doesn't have this flagella, only the motile organism will have this flagella. And this is the structure of flagella, and this flagella is divided into basal body, hook, and the filament. And this is the basal body, and the basal body you can see L, M, and MS ring we can see here. And this L ring is attached or seen in this outer membrane area, and this P ring can be seen in this peptidoglycan region of this bacteria, and this is the M ring. M ring can be seen in the cell membrane, and just above the cell membrane we can see this S ring. And all the four rings are present in gram-negative bacteria, and only S and M ring are present in gram-positive bacteria, indicating that only S and M are required for motility and LP are required for support of the bacteria. And here you can say this is the portion of the bacteria, and it's a short or curved structure connecting filament and the basal body. And it is broader than filament, and it is protein in nature and different from flagellate. And just outer to this hook, you can say this is the filament. It is external to cell and connected to hook at the cell surface. And this filament is made up of flagellate. And this is about the structure of bacterial flagella. So this bacterial flagella is used for the motility or locomotion of bacteria from one place to the another. Next, about the arrangement of flagella. Depending upon the arrangement of the flagella, we can classify bacteria into monotrichous, lopotrichous, apitrichous, and peritrichous. And if the flagella is seen in the one end of the bacteria, we will call it as monotrichous. That means single flagella at one end, we will call it as monotrichous. That is example is Vibrio cholerae. And in lopotrichous, you can see many flagella can be seen in the one portion or one end of the bacteria. We will call it as lopotrichous. Example is Bartonella bacilliformis. And the apitrichous, we can see the many flagella can be seen in both end of the bacteria. That is called apitrichous. The example is Spirulum serpentis. And in peritrichous, we can see this flagella can be seen all over the body of this bacteria, all over the cell of this bacteria. That is called as peritrichous. The example is Escherichia coli. These are about the classification of flagella based upon the location of the flagellum in the bacterial body or bacterial cell. Pili of Fimbria. Pili of Fimbria, it is the hair-like structure of the bacteria. It is arised from the cytoplasm of the gram-negative bacteria. That means only this gram-negative bacteria we can see this pili. Pili is a hair-like structure that we see in the outermost layer of the bacteria, and it is the organ for adhesion. That means the bacteria, by the help of this bacterial pili, the organism will go and bind to the surface of the host. And it is composed of protein called pilin, and it is the length is shorter length. The measurement is 0.1 micron and the breadth is 10 nanometer. You can imagine the size of the pili. So it is shorter in length. It's composed of protein called pilin. This organ for adhesion. That means with the help of this pili, bacteria will go and bind to the host membrane or host glycolipid or glycoprotein area. It will go and bind. And some bacteria will have sex pili. It will help in the transfer of genetic material from one bacteria to the another bacteria. This is about the pili and fimbriae seen in gram-negative bacteria only, not in gram-positive bacteria. Bacterial spore, endospore. It's a highly resistant resting phase, the resting phase of bacteria during abnormal environment. That means this is a resting phase of bacteria. That means when there is a lack of nutrient or high temperature, or if the bacteria is not getting proper nutrition for their growth, what will happen? This bacterial normal vegetative cell will be converted into endospore formation. The bacteria will be converted into endospore. In this endospore formation, due to this endospore formation, the bacterial DNA can be preserved and other cellular organelles can be preserved. And in this condition, the bacteria will be resistant to heat, chemical, radiation, and desiccation at all. That means during this endospore stage of the bacteria can be live longer for years and years. So this endospore will resist heat and chemical or high cold condition. And endospore are called as spores inside the bacteria. That means endo and spore. That means spore inside the bacteria, we will call it as endospore. And one bacteria can seed only one spore. The example are Bacillus species. This is the Bacillus species, Bacillus anthrax, Clostridium, and Sporoparcina species will have these endospores. Structure of endospore. Here you can see this is the structure of endospore. Exosporium, spore cord, next cortex, core wall or spore wall, and later inside you can see this is the core. And the exosporium is made up of lipopolysaccharide and all the organisms, all the small spore-bearing organism doesn't have this exosporium, and some spore-bearing organism will possess this exosporium. And this inner to exosporium, you can see this is the spore cord. The spore cord is made up of keratin-like proteins, and it is impermeable. That means this spore cord will prevent the spore from harmful chemicals. And inner to the spore cord, you can see this is the cortex. This is the thickest layer of this endospore, and it is made up of polysaccharides. This cortex is made up of polysaccharide. And inner to the cortex, you can see this is the core wall. Core wall is also called as spore wall, and this is made up of peptidoglycan layer. Later, this peptidoglycan layer will become cell wall when it will become to vegetative cell. And the innermost layer, you can see this is the core, and it's also called as spore protoplast. And this contains nucleus and also some enzymes such as dipicolic acid. Shape and position of spore, you can see this is the first one is spherical central spore. That means the spherical shaped spore will be seen in the center of the bacterial cell, and it is non-bulging type, and this is oval. Oval central means the spore shape is oval, and it is located in the center of the bacterial cell. And it is oval subterminal. That means the structure of the spore is oval and it is present in the subterminal area. And we can say that these three are non-bulging. That means you can see the spore is seen inside the cell and it is not projected outwards the cells. That is why it is called as non-bulging spores. And next about the bulging spores, we can see that this is a oval subterminal. Same thing only, oval subterminal, but it is bulging. It is oval terminal. The spore will be seen in the terminal portion, but it is bulging. And the spherical terminal means the shape of the spore is spherical and it is present in the terminal portion of the bacterial cell, and also it is bulging type spore. The last one is pre-spore. Pre-spore means the spore is not present inside the bacterial cell, it is seen outside the bacterial cell. Sporulation. Sporulation is a process when there is an unfavorable conditions for the bacteria, mainly the spore-bearing bacteria, that will convert the vegetative cell into spore formation. So this spore formation consists of different steps. In step 1, we can see the axial filament of bacterial DNA is formed in the step 1. When it goes to the step 2, we can see here in the cytoplasmic membrane or plasma membrane, a septa will be seen. That means the septa formation will be occur. From this region, what will happen? The cytoplasm divides and a 4 spore is seen here. Now we have one 4 spore and this one is the mother spore. And the another step, what will happen? This mother spore will engulf this 4 spore. In the third step, we can see here there is a 2-layer of plasma membrane can be seen here. And in the next step, what will happen? The cortex formation can be seen here due to the accumulation of calcium and dihypochloric acid can also be observed here. And in the next step, you can see the protein coat is formed in this 4 spore. And the next step, what will happen? The step 6, we can see the maturation of spore can be occurred in the spore 6. And in the step 7, the liberation of spore from this vegetative cells will be occurred. And these are the cycle of sporulation. First, starting with the step 1, that formation of axial filament of bacterial DNA is formed. Next, the septa will be formed. The septa will be divided. You can get 2 spore, that is 4 spore and mother spore. Later, there is this mother spore, cytoplasm will be engulfed by this 4 spore. In the third step, we can see the 2-layered plasma membrane, and the step 4, the cortex is formed between this plasma membrane, and also the calcium and dihypochloric acid can be observed here. In the 5th step, the protein coat is formed, and in the step 6th step, the maturation of spore will be occur. At last, the liberation of spore will be seen in this last step. Germination of spore. The germination of spore occur when bacterial will get a favorable condition for their growth and metabolism. In artificially, we can do this germination process by heating this spore. While heating this spore, what will happen? This core of this spores will be hydrolyzed. That means the core will be damaged. When the core will be damaged, in the next step, the initiation process will be take place. So what will happen? It will increase the metabolic activity. And again, when we are giving water to these spores, it will release calcium and dihypochloric acid. And after that, the outgrowth will be take place. That means during this outgrowth, cell wall synthesis, protein synthesis, and enzyme activity will be at high level. At last, the spore will be converted into vegetative cell. That means this germination of spore is a reversal process of sporulation. Then resting phase of the spore will be converted into the vegetative phase of this bacteria.
Subscribe to Simply Micromed if you're curious about medical microbiology, infectious diseases, and unraveling the mysteries of tiny organisms. Hit that subscribe button. Join our community of science enthusiasts, and let's explore the microscopic world together. Remember, knowledge is contagious. Spread it by liking and subscribing. Thank you.